Today, we are honored to introduce a study on the bacterial load in meconium. Here are the highlights. 1. The spike-in plasmid method was utilized to perform an analysis on meconium and second-pass feces, yielding both relative and absolute quantitative results. 2. With the absolute quantitative data, the abundance of bacteria in 17 meconium samples and 17 second-pass fecal samples was found to be 1.14 times 10 to the power of 7 copies per gram and 1.59 times 10 to the power of 9 copies per gram respectively. 3. The mode of delivery can significantly influence the alterations and compositions of gut bacteria in a newborn within 72 hours. A total of 17 newborn infants, 11 male and 6 female, were enrolled in this study from May 2023 to June 2023. Among them, 10 were born via Eutosha, and 7 were born via C-section. The average birth weight was 3,300.5 plus or minus 476.2 gram, with an average gestational age of 38.3 plus or minus 1.5 weeks and an average maternal age of 32.2 plus or minus 3.5 years. Meconium samples were collected from all infants within 12 hours of birth. The average abundance of bacteria in 17 meconium samples and second-pass feces was found to be 1.14 times 10 to the power of 7 CFU per gram and 1.59 times 10 to the power of 9 CFU per gram respectively. And the absolute quantification results showed no significant differences in microbial abundance between the Eutosha and C-section groups. For both meconium and second-pass feces, the Bray-Curtis distance-based similarity analysis by PCOA indicated that the meconium samples were clustered separately from the second-pass feces. Following feeding, the distance of each second-pass feces sample increased, indicating that changes in the infant's gut microbiota begin within 72 hours of birth. Based on the mode of delivery, both the meconium and second-pass feces were categorized into Eutosha and C-section groups. The beta diversity analysis revealed no significant difference between the Eutosha and C-section groups in meconium. At the phylum level, there were significant differences between the two groups. The second-pass feces of the vaginal delivery group was mainly composed of 47.6% of proteobacteria, 22.6% of actinobacteria, 20.6% of firmicutes and 9.2% of bacteroidetes, while the C-section group was mainly composed of 58.6% of firmicutes and 40.6% of proteobacteria. The absolute abundance of some phyla, such as bacteroidetes, also decreased in the C-section group. Other phyla, such as actinobacteria, increased their average absolute number 30,506 CFU per gram to 796,5106 CFU per gram, although they did not account for a large percentage of fecal samples from caesarean sections. Summary. In this investigation, we employed the spike-in plasmid method to conduct relative and absolute quantitative analysis of the meconium and the second-pass feces. The abundance of bacteria in meconium samples and second-pass fecal samples was found to be 1.14 times 10 to the power of 7 CFU per gram and 1.59 times 10 to the power of 9 CFU per gram respectively. We determined that the similarity of meconium was unaffected by the mode of delivery, whereas a notable distinction was evident between the second-pass feces of infants delivered via Eutosha and those delivered through C-section. We found an intermediate state between fetuses and adults.